Hello everyone, my name is Eleftherios Garyfallidis. I'm a professor at Intelligent Systems Engineering at Indiana University, and I will talk to you today about the unsupervised tractography segmentation algorithms that we have in Dipy. In the next talk, I will talk to you about supervised tractography segmentation. All right, so in the previous talks, you saw that in Dipy we can generate millions of streamlines, and it can be that a large number of them are invalid, so they do not approximate well the underlying anatomy. And we have this data set, so what do we do with them? What can we do with them? So one thing that we can do is extract bundles, create clusters, and so on. But how do we do that? One of the very important concepts here to understand is the streamlines themselves. The streamlines themselves, they have a data structure that looks like this. So you have here like one point of the streamline and second point, third point, and so on. Here this streamline has 20 points. So streamlines are points connected sequentially together. And they do integrate information along many voxels. Of course, they cross many voxels. They have sequential connectivity information. They can represent shape and orientation. They make good models of bundles if you add many together, better than using surfaces or just points. But there are some known challenges. For example, the order of the points between two streamlines can be the reverse even in the same bundle. What does that mean? Let me get my pointer here. All right, so then that means basically that, look, in this streamline here, the first point starts here on the left, but for this streamline here, even though those have very similar shape and they're close to each other, the point here is the last point, so the first point is here. So we have to be careful, because if we want to figure out some way of an average distance, we have to compare with these points to the closest point, rather than comparing this point to that point. And that creates a computational issues. It's not, there are not many ways to do this efficiently. So and we need that. We need to calculate distances. So we need to calculate distances between streamlines if we want them to cluster them together. Also, another known challenge is there are many points in the streamlines, and the tractograms are big. They can be many gigabytes. There are tractograms now that are even terabytes, and they can be heavy on memory to process them. So we need algorithms to do that fast. So in the past, people were using such metrics, such as the Hausdorff distance, where you would take for each point, you would find the closest points in the other streamline. So for that point, you will find the closest point in this streamline. For this point, you will have that point, and so on. And you do it both from one side from S to S prime, and for S prime to S. And that can be computationally expensive, because if, let's say, a streamline has P points, then just with another streamline, you have to have a complexity that is P squared. But then I introduced this distance, which basically, assuming that the number of points on a streamline are the same, here we have 4 points, 1, 2, 3, 4, and here 1, 2, 3, 4, we can calculate the direct distances shown with here. Those are the Euclidean distances A, B, C, D. And then you can also like flip one of the two streamlines and calculate those distances and keep them. A, B, C, D. So those are, at the end, if you calculate an average distance of those, it will be very different in the direct case than the flipped case. But you have the advantage that this is very efficient, and the complexity now is linear when you calculate these distances. Okay, so now if you use such distances, also it's easy to create centroids. Like let's say if you take the direct version here, then the centroid between these two images is just this average points. If you connect those average points, you create an average streamline, a centroid streamline. So centroids are useful, especially when you do clustering. So using now this concept, we bring forward the minimum average direct and flipped distance. And this is very fast. The equation is written here. So basically that distance is the minimum between the direct, the average direct, and the average flip distance. Now there is a big theoretical advantage using such distance because it is, okay, it is very fast, it's linear type, it's still capsule shape, it's also symmetric, which is really good, and it's a proper distance metric, meaning that it satisfies the triangle inequality and all the other properties that distance metrics should satisfy. And therefore the space, if you're solving an optimization problem, the space is not distorted. You have a nice space to make your calculations. So each streamline needs the same number of points. That's the only negative here. So you have to pre-process your streamlines to have the same number of points. But that one also is done very easily by interpolating the streamline along its length. So yes. Okay. So this is the paper that used quick bundles, a method for tractography simplification, that used this idea of an MDF distance for the purposes of clustering. And one of the big advantages here is that it shows that you do not need to check every single pair of streamlines to calculate a clustering result. So before, like people were trying to take all the pairs of streamlines and create maybe like a big distance matrix, but that is not required if you're using quick bundles. And imagine like if you had a million streamlines and you had, so imagine you will have now to calculate a matrix that here you have 1 million and here you have 1 million streamlines and calculate all the distances between each one of the streamlines. That would be a very, like it will be a very long operation. It will take forever to complete all those pair distances, especially with the data, the modern tractography data sets that are massive. So the answer to this was, okay, we can use a linear time clustering method. And actually, this method was invented for this purpose. I mean, there was no such method before. So meaning that this was a new machine learning algorithm. It was not that we used an existing machine learning algorithm to create that. It was tailor-made, created from scratch to solve this problem of clustering streamlines. And if you see the algorithm, basically, you have the tractogram here, and you have the streamlines coming in, and then the output is a clustering result. And what you do, you have here a for loop that goes through all the n streamlines. So that's happening here. And then you have the areas where you have the evaluation of the direct and flipped distances. So here is where you calculate the MDF distance. And then later here you have, sorry, here, then here you go through the clusters. So you have another for loop that goes through the clusters. So as you go through the streamlines, you also create the clusters dynamically. And the number of clusters is shown here. That's the number of clusters with M. So this is M here is number of clusters. And those are increasing dynamically. Right, so then later at this stage here is an important part where you have to decide if you're going to create a new centroid that will have updates that using the flipped version or the standard version of a streamline, because we have to deal with this ordering problem. Okay. All right. So this is what the algorithm is doing in an abstract fashion, but that's the complete algorithm here expressed. So be happy to spend time to understand what's going on. But here also a visual explanation of the algorithm. So imagine this problem here. You have a bunch of streamlines and you want to find clusters on those streamlines. So now you can basically pick one randomly. Randomness is very important for this method. So you pick one randomly, and then that first becomes the first cluster, and that streamline the first centroid. Then you pick another one. So this now, the one with the red here, this one, you compare it with the existing cluster, and you can see that the distance, the MDF distance between them is 23, but the threshold that we have said is 10. So given that that is bigger, this streamline, the red streamline, will become a new cluster. So this is the new cluster. And then after that, you have the cluster, you have its centroid. Okay, let's now go and pick another streamline. We pick another streamline randomly. We compare now the distance between the first, that streamline, and the centroid of cluster 1. So we compare that and we see, okay, they're quite different. The distance is much bigger than the threshold. So, and then we do the same for the cluster 2. And you can see, okay, now they look similar. The centroid of cluster 2, shown here, looks similar. Right. Good. So then what happens, this red will go in that cluster here, in cluster 2. So then it goes to cluster 2, and then we update the threshold, and then in cluster 2 you will see the centroid will be updated. Okay, let's go now pick another streamline. We pick this streamline here, and we compare it with the centroid of the first cluster. We compare it with the centroid of the second cluster. You can see now here is quite different. So then it's not close to any of those other clusters, so it becomes a third cluster. And then the last streamline, we check it with the first cluster, cluster 1. We check it with a centroid of cluster 2 and of cluster 3, and you can see it's clear that it's very similar to the centroid of cluster 1. So then this is where it will appear. So now we have the final result, and we have all this clustered here, all the clustering results shown here. So now with the colors, you can see also, oh yeah, there are 3 clusters there. They, like, it makes sense, right? Okay. So let's move to the next slide. Oh, I should say this, that notice also that at the end, the centroids are updated, and you can see the different centroids here. That's the final result of the centroids. All right. So here is how quick bundles looks for the corticospinal tract. You can see here the corticospinal tract. This one looks like it goes homogeneously from the bottom here to up here, but actually there are some streamlines here that are diverging, and there are some streamlines here that they stop prematurely. So you can see now, by doing this clustering analysis, we can find those and see them and visualize them. And here are the clusters, and here are the centroids. So you can use them to identify hidden structures of the data and maybe have a better idea about which are the invalid pathways. This, for example, here seems invalid. This one also seems invalid too. It stops too quickly. And you couldn't see that with the current color map here. All right. So that's how quick bundles looks like for the corticospinal tract. Let's look at something else. So here is how quick bundles looks like when it's running, and you can see the streamlines are picked randomly, and then you can see the clusters in the right and the centroids in the middle, and the width of the centroid streamlines is increasing in relation to how many streamlines belong to the corresponding clusters. And at the end, you get the final result, which is shown here on the left. All right, so this is quick bundles in action. And this is quick bundles when you have very large tractograms, and then you get basically, you can get thousands of centroids. If you use our recommended thresholds, which are between 10 millimeters to 20 millimeters, for most data sets, that, especially for human brains, that's what you will get. So here is a summary of quick bundles. Is a fast and supervised way to compute centroids, like a mean description of the bundle, to cluster streamlines to reduce the complexity of the data, simplify your data, using a single threshold that can be in millimeters, for example. If your tractography is in world coordinates, or like in a standard coordinate system, which is in millimeters, then you can use that to identify the best threshold. Quick bundles is not a way to find anatomically described white matter tracks, because those need priors from anatomical studies. Okay, quick bundles was one of the principal methods that inspired the creation of theta machine learning framework. This is very recent work in 2021, and that's a generic machine learning framework, and there is currently an archive paper about it. Also, quick bundles has been used outside of neuroscience. For example, it has been used for clustering GPS orbits and tracks. All right. So then there is also quick bundles X, which is a faster version of quick bundles that basically takes into account the idea that if you have a large threshold, so if you have a large threshold here, then quick bundles is faster and you have less clusters. If you have a small distance threshold, then the number of clusters is high. So what you can do there is basically you can use that and build a divide and conquer technique and create a faster version of quick bundles X. For example, you can start with a high threshold and progressively go to the lower threshold, which is the actual threshold of interest. And that builds these trees, these dynamic trees of bundles. So quick bundles X updates dynamically this tree where you have different layers of thresholds acting on the data. And it can be much faster, even though quick bundles is very fast. With quick bundles X, you can go even faster. For example, clustering 5 million streamlines in 5 minutes. That's with an old computer. And when quick bundles needs 2 hours. Okay. So it is fast, and it is you can use it to make these trees of bundles, and it has low memory usage as well. So high speed, low memory usage. And this is an example of how that looks like when it's running. Here is some random streamlines from the tractogram, and this is how the tree is generated. And then at the end, you have the different layers and the final result. Okay. So Horizon, one of our visualization tools, is using quick bundles under the hood, quick bundles X, and it uses Fury for the visualization, but it uses Dipy for the clustering part, and it allows you to take a tractogram, generate a skeleton of that tractogram, the centroids, then interactively click those centroids and expand the bundles, which then you can edit, as you can see here in this corner. There is a part that we want to remove, so it allows you to remove such parts. So it allows you to interact with the clusters and remove them or include them as needed. All right, so you can use it for selection and refinement of clusters, and then you can save the results as new TRK or other files. So to execute that, you basically write that by Horizon, the tractogram. You can have one or many tractograms included, and you can also have a clustering threshold. And if you specify that, minus cluster here and the clustering threshold, then you can actually see the clusters and interact with them. Otherwise, you get the full tractograms as they are without clusters. Okay, so thank you for your attention. This was the talk about unsupervised learning of tractograms and segmentation in Dipy.